Today we will be talking about the Oxford Nanopore sequencing platform. The Oxford Nanopore platform is a third-generation sequencing approach that utilizes proteins known as nanopores that contain a hollow tube in the core through which molecules can pass. In the Oxford platform, nanopores reside in a synthetic membrane that has a high electrical resistance. The membrane is submerged in an electrophysiological solution, allowing for the generation of an ionic current when a potential is applied across the membrane. The electrical resistant membrane ensures that the current generated only passes through the nanopore. When the DNA or RNA is pulled through the nanopore, the different combinations of nucleotides disrupt the current, and these disruptions can be measured to determine the sequence. Additionally, the sequence is run in real time, allowing for immediate generation of data as the strands pass through the nanopore. The DNA or RNA to be sequenced has two adapters attached, the Y or leader adapter and the HP adapter, on each strand. In addition to adapters, both strands contain an enzyme motor, which allows for each strand to be passed through the nanopore. The membrane contains tether proteins to which the DNA or RNA containing a tether attachment associate in order to draw the DNA or RNA to the nanopore. After tethering, the motor protein attaches briefly with the nanopore while the DNA or RNA is being passed. Once the DNA or RNA is tethered, sequencing starts on the template strand at the 5' end of the Y adapter. As the Y adapter approaches the nanopore, the enzyme motor unwinds the double-stranded DNA so that a single strand is pulled through the nanopore at a time. Once the template strand passes through the nanopore, reaching the HP adapter at the opposite end, the complementary strand can then pass through the nanopore so that both strands are sequenced. After each strand has been sequenced, the enzyme motor can detach from the nanopore to allow the next strand to be sequenced. The Oxford NanoDrop platform has 5 different devices that can be used for sequencing. For all 5 devices, the nanopores will read the entire sequence length passed through the protein. While not available on the market for use yet, SmidgION uses the same nanopore technology as the larger sequencing devices, but is for use with smartphones and other mobile devices. Potential future applications include on-site analysis and remote monitoring. The Flongle, released earlier this year, is an adapter flow cell for use with the MinION and GridION devices. In place of the flow cells that are provided with these devices, Flongle is ideal when sequencing smaller samples, target sequences, single samples, or when MinION gives too much yield. Flongle can also be used to perform rapid quality checks before running larger experiments, as well as for diagnostic purposes. The Flongle is portable when used with MinION, allowing for fast in-field sequencing. Each Flongle contains one flow cell that can provide a total yield of 2 gigabases of data. The MinION is a portable device ideal for sequencing whole genomes of medium size, target sequencing, and metagenomics. As the flow cells hold more channels, it can also be used for multiplexing versus single sequencing if using Flongle. As when used with Flongle, MinION is portable and can be used for in-field sequencing. MinION data can be read by plugging the device into a laptop. MinION contains one flow cell that can provide up to 30 gigabases of data per cell. The GridION is a benchtop sequencing device that allows for up to 5 MinION or Flongle flow cells to be run simultaneously or individually. GridION is ideal for sequencing larger genomes, whole transcriptomes of either cDNA or RNA directly, and for running multiple samples. The device can produce an output of 150 gigabases of data when all 5 cells are simultaneously run. The PromethION is a benchtop sequencing device that allows for either 24 or 48 flow cells to be run at once. This produces a total yield of 4.3 terabases for a 24-cell device and 8.6 terabases for a 48-cell device, as each flow cell contains 3,000 channels. PromethION is ideal for very large genomes, experiments requiring a large number of samples to be run at once, and for population-scale sequencing. All 5 devices are ideal for use in epigenetic research, as nanopore sequencing does not require synthesis or amplification before sequence determination. The pricing on the Oxford Nanopore sequencing machines ranges in cost based on depth of capability. Flongle is cheapest to operate cumulatively between device-based cost, library prep cost, and flow cell cost. PromethION is the most expensive. Library preparation time will vary due to type of sequencing, either whole genome, targeted DNA, or RNA sequencing, as well as specificities such as read length, throughput, identification of fragmentation, among other factors. Error rates will vary by model, but the newest models have 95% single molecule accuracy and 99.99% consensus accuracy. Runtime ranges based on the length of the sequence. For example, 1 minute to 16 hours for the Flongle, 1 minute to 48 hours for the MinION and GridION, and 1 minute to 72 hours for the PromethION. There are many current uses and possible future applications of Oxford Nanopore's technologies. One interesting possible application of the MinION specifically is in response to the use of bioweapons. If the widespread release of bioweapons such as anthrax or plague were to occur, rapid whole genome sequencing of these pathogens would be possible at the site of occurrence with Nanopore. This would allow for characterization of the pathogen and for the detection of any modifications, such as antimicrobial resistance, which would help with containment efforts. Oxford Nanopore sequencing capabilities have also proven useful in developing new therapeutics. One study used Oxford Nanopore whole genome sequencing to help characterize 2 novel bacterial species from an ancient hot spring. These bacteria were shown effective in inhibiting the growth of multiple bacterial species and may be important in deriving new antibiotic compounds. Another useful application of the Oxford Nanopore's MinION was in the sequencing of Lactobacillus plantarum, a probiotic that has proven beneficial but has unwanted side effects in some people. The sequencing of this bacterium has been difficult in the past because of its unusually high number of plasmids. The MinION was used to resolve these plasmids and successfully sequence the bacteria. This information may lead to a better understanding of this probiotic. Illumina systems have a greater number of possible applications and methods than Nanopore technologies, although they share many of the same ones. Illumina has production-scale models that can have a max output of 6,000 gigabases and 20 billion max reads per run. These are great for large and small whole genome sequencing applications as well as entire transcriptomes. Illumina systems have demonstrated error rates for misread bases to be as little as 0.1% in quality studies. Illumina systems are exclusively benchtop equipment and thus limited to the lab. The next-gen sequencing technology is very costly, with sequencing kits started at around $1,000 and the cheapest machine being $20,000. Library prep can take up to 5 hours on some kits with 1.5 hours of hands-on work.